Is it recording? I can't tell. I think it is. So I'm going to do a video. And if it doesn't record, then it didn't record. So in today's video, I would like to discuss the general cell cycle and the role that CDK and cyclin molecules play in regulating that cycle. So we're going to be focusing more on that second aspect of it, the regulation, rather than the actual mechanisms of the cell cycle. And so our general cell cycle you should be familiar with or aware of is this guy right here. Right? So the M is our cell division phase. It's either mitosis or meiosis. In the context of today's lecture, it's more likely mitosis that we're talking about here. G0 is the first phase that exists outside of M phase. Sometimes, under some conditions, G0 is entirely skipped. We go straight to G1. Okay. G1, then S, G2. Collectively, everything except for M is known as interphase. Okay, so we have our cell division phase here in our M, and we have our interphase in all the other steps. Okay. So G0 is our resting phase. This is when the cell is just doing its job. Right? It's existing. If it's a liver cell, it's doing liver things. Right? It's the right size, has the right organelles. It's just chilling, doing its job. Okay. Something happens in that cell's environment that is going to trigger the onset of cell division. Right? And so either this organism needs to grow, or this organism has received some damage or just general wear and tear over time and needs to replace some damaged cells. Right? And so either of those two events are going to release growth factors into the environment of this cell. So that growth factor is either coming from glands that are telling this organism to grow, or from damaged cells nearby that are on their deathbed and are releasing ligands and saying, hey, everybody around me, I'm about to die. We need more of us. You better divide. Okay? So when that growth factor is released, we move out of G0 and into G1. Okay? Now, G1 has a couple different names. It could be gap phase 1, but I like growth phase 1 because it involves growth. Right? And so this is the beginning of the cell cycle for the process of division. And so G1 adds size to the cell, and it starts preparing the actual mechanisms that are going to be utilized in the process of cell division. Once G1 has completed its task, it transitions into S phase, which is the synthesis phase where DNA is replicated. All right, I'm going to be making a couple copies of the cell. I need a couple copies of my DNA. So DNA replication occurs in S phase. When S phase is complete, we move into G2, growth phase 2 or gap phase 2. And this is where we're adding a lot of our extra organelles so that the daughter cells who are created through this process have enough organelles to function. And we finalize the preparations of the mechanics for cell division. Now there are checkpoints between the transition from one phase to the next along the way. So when I go to move from G0 to G1, there's a checkpoint there. Right? The cell is assessing its conditions and making sure that everything is cool to move on. And each of these phases has a checkpoint. Right? And so for G0, that growth factor mentioned before must be present. Okay? And once that growth factor is present, G0 goes through a process of creating a molecule called cyclin. And that cyclin is going to be necessary for the mechanisms of G1 to start up. Right? And so creating the cyclin in G0 starts up the process of G1. Okay? So the cyclin is a molecule. It's going to engage with an enzyme. Okay? It's a family of enzymes called kinases or kinases. And their job is to actually mechanically do things in the cell. They turn on processes. Right? Oftentimes it's through phosphorylation of certain proteins or certain mechanisms, right? But their job is to get things going. Right? And this particular kinase is cyclin-dependent kinase, CDK, because it's dependent on a cyclin in order for it to function. Right? So it has a domain that requires the cyclin be inserted into it so it gets the right shape, to do its job. Collectively, when the cyclin is engaged with the CDK, that overall molecule is called a maturation promoting factor, MPF for short. Okay? So when they're teamed up, they're an MPF. And the MPF is what actually can make things happen as far as the processes go. Right? However, when the cyclin engages with the CDK, that cyclin begins degrading. Right? It begins to fall apart. The rate of degradation is very uniform across these cyclins. Right? And so when the cyclin falls apart entirely, that MPF disengages, it is no longer doing the thing, and that process has come to an end. So this cyclin, by falling apart, is working as a kind of timer for each of these steps. Right? The creation of the cyclin turns the step on, the degradation of the cyclin turns the step off. Okay? Here we have a broken cyclin. Oh, bye. This was my cyclin for G1, so now the G1 stuff can't happen anymore. Right? During the process of G1, I made the cyclin necessary for S phase. Right? And so now I no longer have my G1 cyclin, but I have my S phase cyclin, so it can click into a CDK, make a new MPF, and that MPF will turn on S phase mechanics. Right? Eventually that cyclin degrades, but during S phase, I made a cyclin for G2, right? So forth and so on, perpetuating the cycle. Okay? So here we have a nice, pretty model of that storyline. Right? You make the cyclin, you degrade the cyclin. You make the cyclin, you degrade the cyclin. Works as a timer. Right? And that lifespan, that uniform lifespan of any given cyclin is the timer for that step. Right? So if the G1 cyclin exists for 24 seconds, G1 runs for 24 seconds. Yeah. That's about it. That's the important mechanism. That's the takeaway from this lesson. Right? You have the cyclin CDK combination that's serving as a timer for a cell cycle. It's just an example of the kind of conditions that must be met for a cell cycle to progress beyond checkpoints. There are a lot of other things that the cell cares about. Right? In class, I gave the example of the mitochondria. When a mitochondria is surrounded by an excess of ATP, it goes from being its functional bean shape to being a long tube-like mitochondria shape. This tube shape doesn't do mitochondria things, right? Because it has enough ATP around it already. It doesn't need to make more ATP. So it goes into this resting phase. Right? However, when there's a bunch of mitochondria near each other in this resting phase, they will actually fuse into a nice, long supercomplex. That supercomplex is necessary for generating a particular high-energy molecule that oxidates the environment around it, and that oxidation and that molecule is required for the kinases to work properly. Right? And so that's an example of another way that the cell is regulating the cell division. It's assessing whether or not it has enough ATP in order to do cell division, because cell division is going to cost a lot of energy. Right? And so there's a bunch of stuff like that. S phase has to check to make sure the DNA is good before it moves on. Right? You get the idea. Probably. Hopefully. You better. Okay. Here's just a list of some of the specific cyclins and CDKs that go along with each step. You don't need to know these. It's beyond the scope of the course. But just to give you an idea about the fact that there are multiple different cyclins, multiple different types of CDK. They are utilized in each of the steps, right? But these are like the major ones. So like CDK2 might also be utilized in G2 in minor roles. But like these are the big ones for each. Okay? Is there anything else I needed to say? Nope. I just need to figure out how to stop this recording. And that's it. So let's try that. Let's see. No, I don't want to do that. Back to tab.